Hello everyone, I am Kunika Gupta, a final year PhD student at TIFR Mumbai. Today, under the title Lighting the Cell for a Cause, I'll be discussing one of my PhD projects. Cell is the fundamental unit of life, and if we look at the size chart, we realize that it is so small that it has to be seen under a microscope. A cell has various bioanalytes inside it for its proper functioning, but we are mostly interested in the metal ions, that too my favorite, copper ion. But why am I interested in seeing the copper ions? Copper ions are actually regulated inside the cells for its proper functioning, and they are involved in electron transport mechanisms and neuronal signaling, and there are many others. And any disruption in this homeostasis leads to various pathophysiological conditions, like in the lower concentration, it causes Menkes disease, and in the higher concentration, it causes Wilson's disease. Copper ions also undergo this Fenton's reaction, which leads to the production of reactive oxygen species, leading to inflammation and ultimately causing neurodegenerative diseases. So we have to see the copper ions inside the cell and how are we going to do it. So we plan to develop a fluorescent probe which will selectively bind to the copper ions and start fluorescing. And the mechanism we use here is activity-based sensing, where initially the probe will remain non-fluorescent, and upon binding to the metal ion of our interest, which is copper, it will start giving us fluorescence. And this is very beneficial. It has duality check because once it is binding, and second is the selective reaction. And it is very sensitive because it's a reaction-based strategy. So in my PhD, I have developed this probe where this is the binding site and this is the reporter. And upon reacting with copper, the fluorophore is detached, giving us a high fluorescence. And on the other hand, we have an oxidized copper complex. And we observe that indeed the probe is copper 1 and copper 2 ion selective over all the other biologically relevant metal ions. And we observe this visually as well, that in the cuvette, upon addition of copper ions, we see a bright green fluorescence. So we moved ahead with the live cells where we observe that upon addition of copper ions, there's tremendously high fluorescence which can be observed from the calibration bar as well. So we moved ahead to the zebrafish larvae model, and here we observe that in the presence of inherent copper levels, the fish is well lit. And upon addition of specific copper chelator, the fluorescence decreases, saying that indeed we have developed a copper-specific probe which can see the copper ions inside the cells as well as the live zebrafish model. That's it. Thank you.